Hey, good morning. Taking a look today at section 3.3, we're going to look at operons, which is how genes are able to turn themselves on and off. So this is section 3.3, operons. And an operon is a group of genes and switches. The switches turn those genes on and off. So you don't always need a gene on. You don't always need to be producing certain proteins. And so these operons help to turn those genes off when we don't need them. And then we do need those proteins to turn them back on. So we're going to look at one very specific operon today, the Lac Operon. So Lac Operon works to break down this guy. Hopefully you're recognizing that hexagonal shape. This is lactose. So you can see probably why it's called the Lac Operon. And that hexagonal shape kind of symbolizes that this is a carbohydrate. One I in that word. So lactose is a carbohydrate. Carbohydrates we usually draw as these hexagons. We'll talk more about those later. We have a whole section, a whole chapter on carbohydrates. But for right now, lactose can be poisonous in large amounts. You've probably heard of someone who is lactose intolerant. They're unable to break down lactose, and so when they have lots of it, they get sick. So although it can be poisonous in these larger amounts, it can also be broken down. In fact, we kind of break this bond right here, and we've got two parts, one of them being glucose, which is a usable form of energy. Now I'll go ahead and let you know that the Lac Operon, the one operon that we're looking at, there's hundreds, maybe even millions of different operons. This one is specific to a bacteria cell, a prokaryote. So we, as eukaryotic cells, don't actually have this operon. But we do break down lactose, very similar to how bacteria do. Different protein, though. This is the protein that bacteria use. This is called galactosidase. So again, there's that ASE, lets us know we're working with a protein. What does this one do? Well, it breaks down lactose into one glucose and one galactose. That's actually the other half of lactose, glucose and galactose. So it kind of tells us what it does. Protein breaks things into galactose. So again, it's kind of an enzyme that breaks down lactose into usable parts. And you hopefully can kind of see that the shape of an enzyme is really, really important. That's something you're going to hear a lot over the next couple of weeks. So hopefully you can kind of notice that the lactose gene here, or the lactose carbohydrate, kind of fits right into the galactosidase. And then this kind of acts like an axe and splits that bond between them, just kind of crunches That's how the enzyme works. So those are two of the big players we're looking at today. And we're actually not going to get to them just yet. Skip a few lines. I'm going to come down here. If you really want to be artistic with this, you could draw the double helix, there's kind of wavy areas. But for right now, I'm just going to draw a straight line and hope that you understand that it is that kind of wavy area, because I'm going to call it DNA. And I'm going to say this segment of the DNA here is a gene. It is the Lac Z gene. And the Lac Z gene codes for galactosidase. So that protein we just took a look at, here is its DNA code. We transcribe this into an mRNA message. We send that mRNA message to a ribosome and it gets translated into this protein. But here's the code for it. Here's the Lac Z gene. Over here, we have a promoter. So going back 2 chapters, we talked about promoters. Their job is to attract the RNA polymerase. So this is where the promoter is relative to the gene. He's going to call over the RNA polymerase, get him started, send him on his way, and he's going to transcribe. RNA polymerase is going to transcribe this DNA into mRNA. So nothing new yet. We've seen all this. Just kind of looking at the last chapter and a half all together. Here's the new part. This region in between the promoter and the Lac Z gene, this is called the operator. And the operator by himself does nothing. When it's just the operator there, nothing happens. But what his job to do is to attract the repressor. So just like the promoter attracts RNA polymerase, the operator attracts the repressor. So let's draw the repressor and talk about him real quick. Here is the repressor. And just like the name sounds like, his job is to block transcription. So as long as that repressor is sitting there on the operator, transcription doesn't happen. There is no transcription. RNA polymerase can come here and land all he wants. He's blocked by this repressor. The repressor sitting on the DNA, not allowing transcription to occur. Why is there no transcription occurring? No lactose. Without lactose, there is no need for galactosidase, so no transcription. No lactose, don't waste time, don't waste energy. Why would you want to waste the resources turning this strand of DNA into an mRNA, and then the energy and the resources of turning that mRNA into a protein if you don't need that protein? And that protein's job is to break down lactose. But if you don't have lactose, why make that protein? So here the gene would be turned off. The repressor is blocking transcription, not allowing transcription forward. What happens when there is lactose? And shape, very important. And so perhaps you noticed our repressor, here's the repressor again, same shape. He's being pulled off of the operator. So I'm going to go back down here. I'm going to draw that same series. We have the DNA here. Okay, if you want to get creative with it, you can draw your double helix. I'm just going to draw a line. Here's the Lac Z gene again. Here's the operator, and this time the operator's empty. So the repressor is not sitting in the operator. And here is the promoter again. All right, so all of this is still the same. The difference is that the repressor has been pulled off of the operator. It gets pulled out of there by what we call an inducer. So the inducer binds with the repressor and prevents it from binding with the operator. We call this affinity. So not infinity, but affinity, which basically means love. The repressor likes the operator. It likes being with the operator, likes sitting on the operator. But it loves the inducer. It will leave the operator to go and bind with the inducer. It has a higher affinity, a higher love for the inducer. And that's why it leaves the operator. The path is now clear. The really cool part about all this is that lactose, that substance that we're trying to break down, lactose is the inducer in this system. So you notice the shapes. Again, perhaps you noticed when we drew the repressor that it was built to fit those two hexagons so that they can pull it off. And now that we have lactose, now that lactose is present, the path is clear for transcription. Now our RNA polymerase can land on the promoter, transcribe that gene into mRNA, and have that mRNA go make the galactosidase protein, which is going to break down lactose. How do we know lactose is there? Because it's unblocking transcription. It's binding with the repressor, removing it from the operator, allowing transcription to occur. What happens when that protein does all of its job? When it breaks down all the lactose, we don't have any more lactose. Well, then the repressor is going to return to the operator, shut the system off. No more reason to make that protein to break down that lactose. So this whole system here, just like we started with our definition, a gene and these series of switches that turn the gene on and off, this is an operon. Again, this is just one specific example of an operon, the Lac operon in bacteria cells. Different operons can get a lot more complex, a lot more advanced, but this kind of shows all the basic parts and how they all work together to turn genes on and off. So hopefully that helps for today, and good luck with your studying for the exam next week. We'll see you next time.